Welcome to the cell-cell communication during development module. In this talk, we're going to learn about the concepts of induction and competence. We will first learn about the terms induction, competence, and reciprocal induction, followed by instructive and permissive interactions. We will then define regional specificity and genetic specificity of induction. We're now going to look at the process of induction and competence. Induction are close-range interactions that occur between two or more cells or tissues of different histories and properties, which results in a change in the behavior of certain cells. The inducer is the tissue that produces a signal that changes the cellular behavior of the other tissue or the other cells. This normally occurs to the secretion of factors like paracrine factors that mediate the process of induction. A responder is the cell or tissue that's being induced. So cells in the responding tissue must have the receptor for the inducing factor and the ability to respond to this signal. Finally, competence is the ability to respond to a specific inductive signal. So we're going to look at a few examples where it becomes clearer what these words actually mean. So let's look at what happens during eye development. So the steps that are involved in the eye development are given as follows. The first step is the regions in the brain, which are the neuroectoderm, that bulge out through the process of evagination to form an optic vessel. So if we look at the figure here, we can see that the portion that is shown in pink, which forms a cup-like structure, is that neuroectoderm that has undergone evagination to form an optic vesicle. And this optic vesicle approaches the head ectoderm that is shown in the form of a red line. The optic vesicle secretes paracrine factors called BMP4 and FGF8. The head ectoderm receives the signals from the paracrine factors of BMP4 and FGF8 and is induced to undergo changes to form the lens of the eye. So when we look at the figure, now we can see that red line of that surface ectoderm, a portion of it that is in contact with the optic vesicle has undergone changes to form tissues that will become the future lens of the eye. Thus, the surface ectoderm cells have the competence to form the lens. Now, interestingly, what will happen is the developing lens will in turn secrete paracrine factors that induces the optic vesicle to form the retina. So when we look at the figure, here we can see that the part of the ectoderm that's forming the lens, it will actually secrete paracrine factors that will cause the optic vesicle to now undergo changes and go through the process of induction to form the retina, which is what is shown in the next figure. So, in essence, what I've told you is the optical vesicle induces lens formation, and this is what we saw in the first part of eye development. And then the lens induces the retina formation from the optical vesicle. So each one is inducing each other to form a certain part. And this is called as reciprocal induction. Now let's look at this figure to understand induction and competence better. So when we look at part 1 of the figure, this is where normal induction of the lens is formed by the optic vesicle. Okay, so the optic vesicle is inducing the process of lens formation in the tissues of that surface ectoderm that are present in the head portion of the embryo. Now let us consider a scenario where the optic vesicle is formed not in the head region but in the trunk region. So in the trunk region, we also have an ectoderm, and the optic vesicle has been grafted there, and it is producing the paracrine factors of BMP4 and FGF8. However, the ectoderm is not responding to those inducers, and hence we do not see the formation of a lens. This basically suggests that the ectoderm in the trunk region is not competent, and that is why lens is not being formed in the trunk region. Now let us look at a scenario where the optical vesicle is removed. If the optical vesicle is removed, we do not observe lens formation in the surface ectoderm present in the head. What this tells us is we need the optical vesicle and the inducers that it produces for lens formation to happen. So the lack of inducers does not allow the process of induction to occur. Finally, what we can see is tissues other than the optical vesicle, if they are implanted in the appropriate region, we still don't see any lens formation because no induction occurs. They are not able to make the appropriate inducers that are needed for lens formation. There are two modes of inductive interactions that we can observe during development. The first one is called instructive interactions, where a signal from the inducing cell is necessary for initiating new gene expression in the responding cell. For example, paracrine factors from the optic vesicle are required for head ectoderm to form the lens. So the example of eye development comes in the form of instructive interactions, where a signal from the inducing cell is necessary for initiating new gene expression in the responding cell. The second type of inductive interaction are permissive interactions. In this case, the responding tissue has already been specified. However, it needs only an environment that allows the expression of these traits. For this, let us look at the example of heart development. So, an experiment was performed where scientists removed all cells from a rat cadaver heart using detergents that did not remove the extracellular matrix. So when we look at the figure below and look at panel A, we can see that the red heart is present, and as the heart is being exposed to detergent, it's becoming more and more translucent, indicating that the heart cells have been removed. However, the extracellular matrix is still there that keeps the integrity of the heart. After that, cardiomyocytes were infused into the decellularized heart, and they were monitored over time. What was found was that the cardiomyocytes differentiated to form a contracting recellularized heart. So the heart that was there before basically had no cells when the cardiomyocytes were infused. However, it did have the extracellular matrix. In the picture that is shown on the right side in the blue box, we are seeing the recellularized heart, where the cardiomyocytes have organized themselves and differentiated into the appropriate kind of cells to form a contracting heart, where we can see the kind of contractions in the graphs that are right below the heart. Thus, the environment was permissive in allowing the cardiomyocytes to create contracting heart muscle. So even though there was not one cell present in the decellularized heart to produce any kind of paracrine factors to guide the cardiomyocyte differentiation, the cardiomyocytes were still able to differentiate to form a functional contracting recellularized heart because of the environment of the extracellular matrix. Hence, this is an example of permissive interactions. We're also going to look at another concept which is called regional specificity of induction. Now, in many cells, the epithelial cells have to interact with the adjacent mesenchymal cells, and thus we have epithelial mesenchymal interactions. One of the properties of this interaction is the regional specificity of induction. So let us see what this means in the form of an example. So when we look at feather or claw induction in the case of birds, we observed that in chicks, the epidermis, which is part of the epithelial tissue, secretes proteins that signal the dermis, which is part of the mesenchymal tissue, to form these structures called condensations. The condensed mesenchyme then responds by secreting factors that cause the epidermis to form regional specific cutaneous structures like feathers or claws. So here is an example to show the regional specificity of induction that we just explained in the previous slide. The dermal mesenchyme is responsible for the regional specificity of induction in competent epidermal epithelium. Now, depending from where the mesenchyme was taken, the same epithelium can develop appropriate cutaneous structures. So, when we look at the figure that is shown, we can see that if we take the mesenchyme from the wing area of the bird, and if we associate them with the wing epidermal epithelium, then what we observe is we observe the formation of a wing feather. Now, if we keep the wing epidermal epithelium exactly the same, but change the source of the dermal mesenchyme, so in this case we're using the mesenchyme from the thigh instead of the wing, and we are using the same epidermal epithelium, what we see is we do not see a wing feather made, but we actually see a thigh feather made. And thus we are seeing a different structure being made. Now, if we take the dermal mesenchyme from the foot of the embryo of a bird and mix it or associate it with the wing epidermal epithelium, then what we observe is we don't see feather formation at all, but we see claw formation occur. Thus, even though the epidermal epithelium is the same, the mesenchyme is from a different source, and so depending from where the mesenchyme was taken from, the same epithelium can develop into different types of cutaneous structures, and hence this is an example of regional specificity of induction. Depending on the location of the inducer cells, you can get appropriate responses. Another property of the epithelial mesenchymal interactions is genetic specificity of induction. So in this scenario, what we're going to see is while the mesenchyme may instruct the epithelium as to what sets of genes to activate and what kind of structures to form, the responding epithelium can comply with these instructions only so far as its genome permits. So in order to understand this concept, let's look at the figure. And before we go through the actual experiment, in this particular experiment, we are looking at two different types of organisms, which is a newt and a frog. Now, what we should remember is when we look at the mouth parts of a newt and a frog, in the case of frogs, the frogs in their larval stages, which is the tadpole stages, have these protrusions called as suckers. On the other hand, when we look at the mouth part of a newt, they have these structures in their mouth parts that are called balancers. So remember that the frog tadpole has suckers, whereas the newt has balancers. Okay, now let's look at the experiment. So in this experiment, what they first did is they took a frog embryo that had undergone gastrulation and took a portion of that frog embryo and grafted it into a newt embryo that was in a similar stage of development. So the region was grafted in the area that was the presumptive oral ectoderm, which basically forms the mouth parts. And what they saw is when they allowed that newt embryo to undergo further development, the result was the newt actually had the frog tadpole suckers. So these tadpole suckers came from the part of the frog embryo that was grafted onto the newt gastrula. And so even though the newt gastrula were getting factors that were secreted by the frog tissues, they still could not undergo induction to form the balancers that we should have seen in the newts. they actually formed the frog tadpole suckers because that is what the genome specifies since they were of frog origin. Now let's look at the converse scenario where we have the newt gastrula's tissues from the embryo grafted onto a frog gastrula in the area that forms the oral ectoderm. When we allow this frog embryo to develop and look at the tadpole now does not show the actual suckers that is seen on the frog tadpoles. It shows the presence of balancers that are seen in the newts. So this is the part that arose from the cells that were grafted from the newt gastrula. So even though the newt gastrula obtained all the induction signals from the appropriate cells in the frog gastrula, its genome cannot specify the formation of suckers. It can only specify the formation of balancers. And thus these are examples of the genetic specificity of induction. One thing to keep in mind is the instructions of these mesenchymal tissues can actually be cross-species. So whatever signals was obtained from a frog, a newt can also respond to it and vice versa.
With this, we come to the end of our talk where we learned about the concepts of induction, competence, and reciprocal induction. We also learned about the different types of inductive interactions, which include instructive interactions and permissive interactions. We then learned about the regional specificity of induction as well as genetic specificity of induction.